Hey, it's Mr. Shrum. I think something happened with the end of that last recording, so I'm going to go over this slide again. We were talking about donating blood and blood transfusions and who could get what blood. And during certain surgeries, and if someone is injured and they lose a lot of blood, they may need a blood transfusion. And considering what type of blood they have and what donor they should receive is very important. The O blood type is considered to be a universal donor because a person with any blood type can receive O. However, people with the O blood type can receive blood from only those with the same blood type, so they need O. Why do you think this is so? What blood type would be the universal recipient? So instead of the universal donor, which they could give blood to everybody, who's the person that could receive blood from any blood type? Well, blood type O doesn't have any antigens, so it doesn't trigger an immune response in humans with other blood types. However, people with type O blood can receive only O blood because other blood types all contain antigens, which would trigger an immune response. People with AB blood are universal recipients because they have both A and B antigens. So they can get blood from A, B, AB, and O. Polygenic traits. There are traits in our body that are controlled by genes. These traits are called polygenic traits. Each gene makes a contribution to the expression of the character. Traits such as height, eye color, and skin tone in humans are examples of polygenic inheritance. Brown eye color is a dominant trait. However, several genes determine the eye color phenotype. That's why we see varying shades of eye color in people. If you look over here, you see bluish, darkish gray with a golden circle around the pupil. We have brown eyes, maybe hazel. These are hazel, kind of greenish, yellowish eyes. And then blue, brighter blue with a little gold ring around the pupil. Almost turquoise blue, maybe not turquoise, less green, but very light blue, maybe a little green. And then dark brown, well, not dark brown, but a nice chocolatey brown. Very cool. Anyway, we were referring to sex-linked traits earlier, and the X and Y chromosomes determine the sex of an organism. We call these sex chromosomes. Every individual has a pair of sex chromosomes, one from each parent. Females have a set of X chromosomes. Males have XY chromosomes. That means the father determines the sex of the baby. Certain genes called sex-linked genes are present in sex chromosomes. The traits encoded by these genes are sex-linked traits. Watch the video on the next screen for an explanation of the sex-linked traits. So I'll share the computer sound with you. Let's watch.
It's a fact of life. Males and females are different. One of the main reasons they're different is because they have different chromosomes. Males have XY chromosomes, and females have XX chromosomes. Traits that appear on the X chromosome are called X-linked traits. Traits that appear on the Y chromosome are called Y-linked traits. Genetic traits are passed from parent to offspring through these chromosomes, and both sexes express different traits because of differences in their sex chromosomes. Men inherit only one X chromosome, while women will inherit two. Because male sex is determined by the presence of a Y chromosome, females do not inherit a Y chromosome from their fathers. A particular type of gene can have several variations in its DNA sequence. These different variations are called alleles. Some alleles are dominant and suppress recessive alleles. This means that recessive traits need matching alleles, one from each parent. X-linked recessive traits cannot be expressed if there is a matching dominant allele. This means that women are more likely to be carriers of recessive traits. Let's look at an example of an X-linked recessive trait, colorblindness. The genes that affect how our eyes perceive colors are present on the X chromosome. Red-green colorblindness is caused by a mutation on a recessive gene on the X chromosome. So, women who receive two X chromosomes can carry this mutated gene and not be affected by it. Men who inherit the mutated gene, however, are more likely to be affected by it. Here's why. If a father is colorblind, he has the recessive gene on his X chromosome. If the father has a son, the child won't have colorblindness because the father will pass on his Y chromosome. If the father has a girl, she will be a carrier for the genetic mutation. She will receive his X chromosome, but she won't be colorblind because she receives another dominant gene on the X chromosome from her mother. If a mother is a carrier, she has a 50-50 chance of passing on the genetic mutation to her daughter because she donates only one out of two X chromosomes to her daughter. This daughter will be a carrier, but she will not be colorblind. The mother also has a 50-50 shot of passing the gene on to her son. The son who inherits the gene will be colorblind. For X-linked dominant traits, only one dominant gene needs to be inherited for the trait to be expressed because dominant traits overpower recessive traits. This means that a male or female offspring who inherits one dominant allele from either parent will express the trait. An example of an X-linked dominant trait is Golts Syndrome, a rare disorder that causes skin lesions and birth defects. Golts Syndrome is a dominant trait carried on the X chromosome. It can be passed to female offspring on the X chromosome donated from the father. Because males inherit the Y chromosome from their father, sons will not inherit the condition if the father is affected. If the mother is affected, the disease can be passed to the child on the mother's X chromosome. Because both sexes have X chromosomes, only an affected mother can pass the trait on to a male offspring. Y chromosomes are different. All Y-linked traits are expressed on the Y chromosome. The Y chromosome is smaller than the X chromosome, so fewer genes are Y-linked. Y-linked infertility is one example of a Y-linked trait caused by a gene on the Y chromosome that controls sperm production. With the help of medical science, fathers with this trait can sometimes conceive children. But because the trait is passed on the Y chromosome, the son will inherit it from the father. Genes, which code for all our different traits, make up about 1% of our genetic code. However, remember that all sex-linked traits passed from parent to child travel on either the X or the Y chromosome. And that's why we have so much in common with our parents.
Yeah, so that was a nice little rundown of X-linked and Y-linked chromosomes. So we're going to look at a lesson activity about hemophilia. Hemophilia is a sex-linked genetic disorder that inhibits the blood's ability to clot properly. The image shows how hemophilia passes on from parent to child. Study the image and then answer the questions that follow. X-linked inheritance hemophilia. So here we go. Affected father is red, unaffected mother is green. So green is okay, red is affected. And we have an unaffected father and a carrier mother. Okay, so we see that part A, is the gene that causes hemophilia recessive or dominant? Use the image to explain your reasoning. So we, from this, it is recessive, okay? Because the affected father, none of the offspring is affected. So that means it is a recessive trait. The daughters get one X from their mother, one X from their father, and they are carriers, but they are not affected. So it's a recessive gene. However, in the second case, where it's tied to the X chromosome, because the sons need only one X chromosome from their mother, and they only receive a Y from the father. This leads to a son being affected because they only have one X chromosome. They get it from the mother, 50% chance, or 25% chance, because it's 50% sons and then half of the sons get affected. And then a daughter is a carrier, much like the same thing as over here. Get one X from the mother, one X from the father, and one daughter is a carrier. So we say that is recessive. The mother who is a carrier is heterozygous. Heterozygous means a carrier, but she does not have the disease. So she has it, but she does not have the disease. She's just a carrier. That's recessive. The only offspring who have the disease are the boys who inherited one mutated gene. All right, choose one of the offspring from the image and decide on your own the genotype of his or her partner. Then draw a pedigree chart showing the possible genotypes of their offspring. Make sure your pedigree chart shows which individuals are homozygous and heterozygous for the hemophilia gene. The chart should show all possibilities for the offspring. And here's an example. We have an affected male, an unaffected, sorry about that, an affected male and a carrier female. So heterozygous, she has the trait, but she is not demonstrating. She does not have the disease. She's just a carrier. So someone with a carrier, someone carrying the recessive gene, and someone affected have kids. One person is female and homozygous recessive. So they got both recessive genes from their parents, and she is affected. One female is heterozygous, just a carrier. One male is normal, unaffected, and then one male is affected again. So this is worst case scenario. A carrier person, well, not worst case, I guess worst case would be both parents affected, but this is pretty bad too. Part C, calculate the percent probability that a male or a female offspring will be a carrier, be an unaffected non-carrier, or have the disease. A female offspring has a 50% chance of having the disease and a 50% chance of being a carrier. Male children have a 50% chance of having the disease and 50% chance of being an unaffected non-carrier. So yeah, that checks out with the above case. And this one too. So female, 50%. 50% being a carrier, and then 50% females being affected, dominant, homozygous, recessive. And then you have 50% males being unaffected, and then 50% being affected by the disease. Part D, hemophilia, is a negative genetic mutation because it causes a serious disease. Why do you think this gene mutation still persists in our genome? Well, it's a recessive trait, so women can be carriers and not know about it. Also, males who have the disease can still pass on the genes to their daughters, making them carriers. So that's how it keeps it going. All right, so moving on to the final section, genetic research. Genetic research is a newer and exciting field in science. Understanding the genome helps us understand the origins of certain diseases. The genome also explains how different genes affect different body functions. Let's take a look at an outstanding program that revolutionized the world of genetics, known as the Human Genome Project. The Human Genome Project was initiated to study the DNA sequence in humans and identify the genes that make up our DNA. The entire set of genes present in an organism is called its genome. The project focused on the human genome, determining the base pairs and the genes that make up human DNA. Human DNA has about 3 billion base pairs, making it complex to analyze. The project took 13 years from 1990 to 2003 to complete. Back in 1986, a conference held in Santa Fe, New Mexico, discussed the details of analyzing the human genome. There were a number of reasons for initiating the project. US scientists thought that analyzing the genome would provide insights into the causes and treatments for cancer. The US Department of Energy wanted to study permanent damage in DNA caused by long-term exposure to radiation. This interest led to the development of the project. Based on the data gathered during the project, researchers have estimated that there are probably about 20,500 human genes. The project has also provided information on the structure, organization, and function of the complete set of human genes. This information helps detect genes that can cause inherited diseases and also identify major genetic abnormalities in cancer. The Human Genome Project was a breakthrough in genetic research. It provided a lot of insight into the human genome and opened doors for further research. Genetic research is important to many other scientific fields as well. So we're going to take a look at some of them. In the realm of agriculture, scientists have developed genetically modified crops by altering genetic material, and they can remove or manipulate existing genes in crops to improve their resistance to diseases or increase their yield. These modifications may help the crops grow in different environments, thus increasing the food supply. As a step toward the future, genetic modifications may also help engineer crops that have an increased shelf life. GMOs are, how do we say this? Somewhat controversial. On the other hand, with medicine, it allows scientists to find the probability of a person inheriting a particular disease. Mutations in a person's DNA can determine a genetic disorder, and treatment of somatic mutations that develop in a person's lifetime, such as cancer, is also possible with the help of genetic research. So in the realm of medicine, it is actually pretty beneficial. GMOs, many GMOs involve crops and whether food produced from them is safe and what impact growing them will have on the environment. There's just a lot of controversy surrounding GMOs, especially with regard to their release outside of lab environments. There's been a lot of litigation, suing, international trade disputes, protests, and regulation of commercial products in some countries. A lot of people are interested if they provoke an allergic reaction in some people, whether genes could transfer to human cells, whether genes not approved for human consumption could outcross into the food supply. Each food needs to be tested on a case-by-case basis. Some would argue some of the genes could increase herbicide resistance. So I just say do more research. Conservation. Some species of organisms are endangered because of destruction of their habitats. Studying the genes of endangered and threatened species can help scientists guarantee survival in the future. Genetic variation and genetic diversity are closely related. Genetic variation and genetic, excuse me. Genetic variation is the difference in genes among organisms of a species. Genetic diversity is the amount of genetic variation across the entire species. This differs from population to population, and it allows a species to survive in a changing environment. Low genetic diversity is an indication that the particular species will not be able to adapt to a change in its environment. Genetic research can help such species survive in extreme conditions. Yes, changing the whole infrastructure of the world would be harder. That would probably save endangered species, but I guess this sounds cooler to scientists. I'm not sure where that's coming from. Geneticists study DNA found in the fossils of past species and compare it with the DNA of present species to learn how they are linked. Comparing genomes can help us find evolutionary links between species, even between current and extinct species. So there's a video, and I'll post that in the section, but that's it for inheritance patterns and genetic research. If you have any more questions, let me know, and I will help you out as best I can. Until then, see you guys. Bye-bye.